Hello, class. This is your first lecture, and I really want to go over it because there are some major ways that you can study this course. So not only can you do well, but I think you can put a lot of these facts together. Many times biology is taught, so you have this fact, that fact, and this fact, and at the end of the time, everybody, including myself, when I first took biology, is brain dead, didn't know where everything worked. So in this situation here, what we're doing, I've put together some ways in which you can study. We're going to go through concept maps, flow diagrams, and feedback loops. You can read it as well, but I think this is key for you as you go through this course. So let me stop it right there. So what you're seeing right here, up over here, this is the concept maps that has 1, 2, 3, 4 concepts. Every one of your chapters has 4 concepts, no more. And each one of those 4 concepts is shown in your chapter. Of these concepts, there is one that we break up into a flow diagram that goes like this. And of these flow diagram brackets, we have one that ends up being a feedback loop. I'm going to go through all these in a minute. So, concept maps, sub-concept maps, flow diagrams, and feedback loops. Why will these tools help? Well, the reason these tools will help is because they basically give you an idea of how I think. I know that seems dangerous, but what they do is they give you a conceptual approach of how to study biology so that it's put together in a way that you can see what the questions are and what they're not. You can read that text in a minute by yourself. So let's begin with the concept maps. So the concept map is right here. And so every one of your chapters has a title. And that chapter then is broken up into these component parts. So when you're studying for an exam, you're saying, how should I study for the exam? You start right here and see what's in that chapter. Because I put that chapter together with these 4 major points in there, and each one of these major points is going to have questions, right? But there are only 4 concepts, and many chapters have hundreds of concepts, and the student doesn't know what to study for. So concept maps is your guide to the chapter. So when you're reading the chapter, you're saying, okay, he's really telling me that there's something I got to know about the scientific method. I better know something about the non-scientific method. There's something about ethics he wants me to know, and there's something about research. All of these are the major ideas. So you have in your brain what are the important aspects to approach a chapter. So it's not just reading the chapters. What are the major points? Okay, what do I have to get from the major points? Okay, so then as we continue to scroll down here, okay, I'll clear that up in a minute. We're going to go past this here in a second and come to this. I clear that. And like I said earlier, you have a general concept and then you have the sub-concept. And these sub-concepts are found in your chapter. And so it usually has a scientific method, requires, and it has something over here that is really very important. So you have a major concept here, and then the sub-concept in each one of your chapters. From this, we now move on to one of my favorites, which are flow diagrams. Now, let me get to the point first by using this program, and then I'll talk to you about what a flow diagram is. A flow diagram basically is we're taking one of the concepts there and we're breaking it up into A leads to B, B to C, and C to D. Okay, that's a flow diagram. All right, A to B to C to D. And that's what each one of these will go into depth in a minute. But a flow diagram is, you can even think of it as simple as you get out of bed, you go wash your teeth, you go eat your breakfast, you get into your Maserati as you take off for your position as CEO of IBM in San Diego or something like that. That's a flow diagram. That's a process. And biology is nothing more than process. Everything we do, one thing leads to another, to another, to another. So let me show you this in a minute. And that's our flow diagram. It will be coming up here. Still trying to learn how to use this program a little bit better, but we're going to get there. And here's our flow diagram. So the flow diagram basically shows, so I clear this, the preliminary, this is a scientific method, it's mentioned in one of your chapters. Preliminary experiments lead to a hypothesis, which means that there has to be an ethical evaluation of that hypothesis, which means that there have to be experiments, which means there has to be analysis and conclusions, and you either accept or reject the hypothesis. You see how that flows? Rather than going out for days, when you're studying for an exam, you're saying, what's he going to ask me about? Can you see this? I want to say, after the hypothesis has been designed by you, whatever, what are some of the considerations that need to be followed next, or something on that order. But this is your backbone for studying what's going on within each section, how we get oxygen, how our heart controls, or how our heart rate is controlled, how we make ATP. It's all based upon flow diagrams. If you get these flow diagrams down, you can answer any question I have to give you. And I'll show you something here in a minute that makes it a little bit easier. So flow diagrams are just super important. Alicia and I have spent the entire, actually 8 months redoing a lot of these flow diagrams, and we're not through yet. So if you have comments, something's not clear, let us know. All right, this is my way of communicating to you about how you should think about what you're studying. All right, continuing on, if you take a look here, and we'll just wait as the system goes, right? So the general outline of a flow diagram, and Alicia's put this in here in a nice way, is it's a sequence of actions that lead to a goal. Can be you playing football, can be you playing volleyball, can be you setting up to take over the world. And then you talk about it in terms of the sequence. It's really the sequence that I want you to get down, and the specific steps that increase heart rate. So it's just not heart rate anymore. You know the specific steps. It could be global warming. It could be anything. And then the output. Flow diagrams really lead to feedback loops. So feedback loops refer to, if I can do this, here we go again, a process that's just a little bit more conceptual. And I think we've tried to really make these feedback loops something that you can appreciate. So here's our flow diagram. From our flow diagram, there might be something in this box that I really want to make a flow diagram out of it. Excuse me. From the flow diagram here, I want to make a feedback loop, which is here. So this is the feedback loop here, this flow diagram there. Excuse me for misspeaking, I get too excited. So then we go on down a little bit more. Let me clear this up again. And as we begin to look at this, we'll get to a feedback loop here in a second. I think I've explained the flow diagram. So here we go. Let me just bring it up a little bit more. Perfect. Feedback loop is this. You're going to have the system is going to have a certain set point, meaning it's going to have, in this case, let's say your temperature should be at 98.6 degrees. All right, so that's the set point, and that's what the output should be. All right, but wait a minute. What makes that 98.6 are all these effectors. Okay, it could be exercise, it could be metabolism, it could be a whole bunch of things. So this effector here produces the output. And then this output is picked up by a receptor which sends the information back to the comparator. The comparator does this, class. It takes whatever the set point says and says, well, the set point wants it to be at 98.6 degrees Fahrenheit. And the receptor is saying it's at 98.6. Well, let's make it interesting. 98.0. So it's a little bit cooler. So the comparator is going to say, we got to be at 98.6. So the comparator is going to have the effectors continue to work so that this temperature will rise. If the comparator has the effector continue to work, it's a positive feedback. Let's stop for a minute. 98.6 is what we wanted, what the set point was going to be. The output was 98. The receptors say 98. The comparator says, wait a minute, we got to be at 98.6. So it'll cause the effectors here to continue to work till it gets to 98.6. If you take a look at our feedback loops, what we did, because this can be super confusing, we put number 1 is a set point. Number 2 is the effector. Number 3 is the output. Number 4 is the receptor down here. And number 5 is a comparator. Okay, this comparator messes up everybody, but what class you got to understand, what's key is biological systems do things, but they do it relative to a set point, whether it's temperature, heart rate, or whatever. Biological systems are set up to have effectors that cause outputs, and those outputs have to be continuously looked at. All right, for me to talk like this, the output is being monitored by these receptors here that's sending signals back to the comparator and the set point, which are in the brain, and then the effectors, my mouth, the larynx, the lungs, all that work. This is how we think in biology, and it's right there. So let me clear that up just a minute. I'll give you another example here with this one right below it, which is in our first chapter. The scientist wants reliable data, okay? All right, so the output over here has to be that what the data is over here is, quote, reliable. All right, and the way you understand that is that data comes from the receptors, goes back to the comparator, and to see whether or not the data is reliable. But you know, this by itself is not as meaningful as if I said reliable data means that I want 5 pages or whatever, something quantifiable, right? Over here, I have 7 pages of data, all right? The receptor is going to say 7 pages. The comparator is going to say, it's supposed to be 5. We already got 7. Tell you what, boys, we don't need any more. And so it shuts it off, and it becomes, therefore, a negative feedback loop, all right? The course, each chapter has a flow diagram and a feedback loop. And the flow diagram and the feedback loop are key for your extra credit paper. I'll put that down here right now. Your extra credit paper needs to have in it a feedback loop and a flow diagram. And I do know most of you have not been taught how to think that way. It's no reflection on teachers or anybody, but I'm trying to give many of you who are just going to take this one course in biology and that's it, a conceptual way of looking at things. It is not just memorization of facts. It's how to put things together with the flow diagram and the feedback loops. And so each one of these points is listed right here. And there's a real interesting one that I want to bring up, which is down here, which is no feedback loop. Okay, what are you talking about? So let's go down here. And I'll bring it up and show you what I'm talking about. One little bit more. Right here. If you take a look right here, you can see we have a set point, we have a comparator, so we're going to go 1, 2, 3, and 4. Right? Guess what? We're missing the receptor. Let's take a baseball hitter. He gets the ball, or he sees the ball coming, and he swings at the ball. A person who is swinging at a baseball, does he or she have time for a receptor signal to come this way back to the comparator and say, well, wait a minute, you better change. It's not going to be a curveball. It's going to be a slider or something. All right. The problem is in many systems, even in biology, there's no time for this to occur. So there's no time for this to occur. It's gone. And so the set point's going to say, hit the ball. And by hit the ball, it's going to say to the muscles, all right, biceps do this, leg muscles do this, etc., etc. And then you're going to try and see if you can hit the ball. All right? This happens in boxing. It happens in kick fighting. It happens in any system which is very fast and where the brain doesn't have time to compare. These are called ballistic movements. And it's right there. And it's a fascinating area. Ballistic movements occur in biology And this whole idea of feedback loops has another ramification. You can see the set point, comparator, effector, and output, and even the receptor. They all work. What happens if they don't work? What happens if normally you have a receptor down here? It goes like this when you walk. Clear that. All right, so you have a receptor that says walk and tells the comparator how fast you walk, and set point says walk. What happens if that's gone? What happens if all of a sudden the receptor's gone and you're supposed to walk? Are you going to be able to walk as smoothly? No, you're not. So a lot of the pathologies in biology deal with something wrong either with the receptors, effectors, or set point. And we're going to talk more about that later. It's going to put it all together for you in a nice way if you embrace it and go through it through the course. I think those are the major points I wanted to bring out so that you can understand what's going on. And I want to thank you for your time and attention. So remember, we have concept maps that there's one in each chapter, and each concept map has 4 concepts. So you have your concept map from which you have 4 subconcepts, and that's what you study for. That's what you're saying. That's what you're saying. Okay, this is what he wants me to know. And then in each chapter, the flow diagram, understand and master it, and then there's the feedback loop. And again, in these areas here, if you have questions or something, you can say, hey, I don't understand this. Would you go over this or do something? And I'd be happy to do it, because I think if you learn it this way, you'll learn it in a positive way, and it'll help you in all your other courses as well. I know that's a big claim, but I think it's true. All right, have a good day. Thank you very much.